Welcome, everyone. Thank you for joining us for our presentation titled Tips for Successful Single-Cell and Single-Nucleus RNA-Seq. It's my pleasure to introduce you to Luciano Martelotto, Scientific Director, Single Cell Core Laboratory at Harvard Medical School. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following the end of the presentation. Please join me in welcoming Luciano Martelotto. I will now turn the presentation over to him.
Hello, everyone. For those that don't know me, my name is Luciano. I work at Harvard Medical School at the Single Cell Core Laboratory. And it's my pleasure today to, give you a full list of tips for single cell experiments. This is what I like to call my notes from the lab. Just as a full disclosure, these are my understanding of of the single cell field, the things that I've collected over the years that I've been doing this. And I just put them on a series of slides, and I would like to share those with you, mostly for those that are just starting in the single cell field. I think this is going to be quite useful. So the talk is divided into two sessions. The part A is I'm going to try to cover a series of topics here that are blacked out, and hopefully I can cover that within 50 minutes or so, and then we're going to stop, and then we move to the next session, which is after this session finishes in about an hour. Okay, so but essentially these are all the topics that I would like to cover today. If we manage to have time, I will leave a few minutes at the end to have a few questions. So one of the things that the reason why I started doing biology or single-cell genomics is when I actually realized that we have so many cells in our body. There are nearly 40 trillion cells in our body, which, and after the application of single cells, we were able to identify, roughly around 200 cells. And I think the more single-cell experiments we do, and the more atlases we start building, the more we realize that we have actually more cell types. And in the past, most of these studies have been done when we were doing genomics or transcriptomics. Most of these have been done using bulk analysis, that is taking the RNA or the DNA from hundreds of millions of cells at once. So, and obviously when you do that, it's a very informative approach, and it helps us understand a lot of the genomics and transcriptomics of the human and also from animal models. But what we are getting from that is an average signal. So essentially, what you're getting is a signal that contains everything that is in there. Usually, is the model population, or the population is more abundant. But what we wanted to do is actually understand what is the actual granularity of the data. How many cells do we have there? Are all the cells the same or different? And the reason why we were not doing that, it was because it was a technical problem here. And the problem was that we don't have sufficient material in a single cell to actually to apply it in the past, the method that we were doing to actually detect, a mutation or detect a transcript or differences in transcription. So, and this is in the last 13 years, I would say, there's been an enormous progress in the understanding of how to actually tackle the technical hurdle. And that's when the single-cell genomic field started to build up. Sorry, I'm trying to, okay. So as I mentioned before, we have nearly 40 trillion cells, and every single cell has around 6 billion DNA bases. And this is in every single cell. And if you consider all the epigenetic modification, that makes the complexity of every cell even bigger. So at any given time, this is just roughly, you may have around 50,000 molecules of RNA, and only nearly less than 1% is in the nucleus, and the majority of the RNA is in the cytosol. And in this case, is taking into account not all the genes are expressed at the same time, and you have a lot of transcriptional variants, and in this case are splice variants, for example. And all of those transcripts are translated into thousands of proteins. And if you consider our immune cells, for example, they have their unique repertoire of antibodies, which make the complexity of a particular tissue extremely, extremely high. That's what we want to do, is just to move from the average resolution that we have in the past, in which we had a tissue and we were able to dissect the tissue and observe under the microscope. And then from there, we move to something in which we actually get an average signal. So we take a sample, we do the RNA-Seq or DNA-Seq, and we get an average signal from that. And we want to move from something like that to something in which we actually can resolve all the cells that this particular tissue and all the different components and all the different cell types that actually form in that particular tissue. So in a way, what we want to do, for example, if you have a disease like a tumor case, like a tumor in this case, you don't only have tumor cells. So what you have is an ecosystem. And that ecosystem will consist in tumor cells and in normal cells, and or the stroma cells, and then you have immune infiltrate that are trying to target the tumor just to get rid of it, and you have endothelial cells and all of that are trying to kind of feed the tumor, or that's the system that the tumor hijacked to actually survive. So what we want to do is to be able to resolve those cell populations and then use computational analysis to resolve potential differential expression between the cell and see what things are different between the tumor and the normal cells. Now, obviously, when we look at the tumor, you probably have seen this slide many times before. But what I like about this slide is because for the non-expert in heterogeneity or on single cell, it sort of put a picture of how we can interpret how the different technologies have been adding to this disentangling of the complexity of a particular tissue. So imagine a complex tissue as a bowl of fruits, right? So you can, but then you can apply different technologies and get different resolutions. So for example, when we were doing bulk genomics, which is what I was mentioning before, we will be able to we can compare that as a preparing and smoothing. So you can get a zip of that smoothing and say, okay, well, I think here maybe we have strawberry, we may have some pineapple, and maybe have some banana. But you don't know exactly in what proportion you actually have those things in there, right? But then if you combine that with other powerful techniques like a flow cytometry and you can, which means you can actually separate all the different components of the individual components into different categories and all the different fruit and prepare different smoothings. And then you can say, okay, well, I have all of these. This is pineapple smoothing. This is a strawberry smoothing. Whatever. So, but that's good. But again, we know that within that, especially within the particular tissue, not all the cells are composing, not all the epithelial cells are the same. So when we combine that with single-cell genomics and flow cytometry, we are able to understand how much differences or what the differences are within a particular cell type, right? And that's what this is trying to sort of emphasize. Now, what is more exciting is that now we are moving to from single cell space in which we actually take a tissue, we dissociate it in all the single cells, and then we analyze that into single cell genomics or single cell transcriptomics. But we lose an important component of that, which is the physical location of that particular cell. And that is something that we're not going to talk today, but this is the field of spatial transcriptomics, which allows you to essentially see the position, not just only the particular cell or the particular components of that tissue, but it also tells you exactly where that component was located and what's the proximity with other cells and how they may be interacting. And you can sort of see all the predictions that you get from single cell genomics to see whether that pans out in the actual tissue. Okay, so this is, as I say, this is a very introductory sort of a few slides just to understand how complex humans how complex tissues in general, not just only humans, are and what we are trying to do single cell genomics, and why we are evolving to single cell or spatial transcriptomics. Now, one of the main reasons why we do single cell genomics is because the biology of the systems are really complex. So we have a lot of tissue heterogeneity. Here is an example of a series of Disney plots in which every single dot will represent a particular cell, and all the colors represent a cell type. So what you can see some cell types that are represented in every single one of the tissue, but the composition and the enumeration of that may be different. And some cell types are particular for a particular tissue or specific for a particular tissue. So the tissues and the organs are very, very complex, and we cannot really assume that all the cell composition of all the tissues is the same. So that's why this is something that obviously became more obvious when we started doing the atlases of all these tissues. Now, there is a lot of cell heterogeneity. So, for example, if you take the cell cycle, all right, so you said the cells are not all, they are not all the cells. If you take a tissue and you dissociate the cells into individual cells, not all the cells are in the same state, all right? So some cells are dividing, some cells are arrested, some cells are differentiated. So the cells are actually continuously moving in this internal clock that is the cell cycle. So there is a heterogeneity in there. Now, in a tissue, there are cells that are dying, cells that are alive, and cells that are already dead. All right. So this is also a very important aspect of heterogeneity within the cell. There is a regulatory network. So all the cells interact with each other. It's not that every single cell is a single entity. This is a tissue. This is a system. Every single system has a working towards an ultimate goal, which is survival. Then we have gene relationships, all right? So the expression of one gene alters the expression of another gene. A mutation in one particular gene is only going to be effective if there is a mutation in another gene, okay? And you get all these epistatic interactions between genes. So that's happened and can happen within tissues, and especially within disease. And that it's something that you can only see at the level of the single cells. And you have clonotypes. For example, you can, in the case of the immune cells, you have, different clonotypes, and that is based on the VDJ recombination of the TCR, for example, in this case that I'm showing here, and that generates a lot of heterogeneity in terms. So maybe all of those are T cells, but they are different clones. They are supposed to target different part of the antigen or different antigens, and some of them are not yet targeting anything, right? And then also, if you look at the cells in a differentiation path, they may be actually located along a differentiation path, and that is also gives a little bit of an extra complexity or heterogeneity to the system. So as you can see, depending on how you see this system, you can actually it can get very, very, very complex. So now, if you look at the organism level, you can see that this is two examples: an example of frog and an example of zebrafish, in which, for example, you can take different samples. You take the different the embryo at the different stages, and then you do atlases of all the cells involved in that, and you can see how there is a lot of you can actually track the evolution of that particular embryo from very mature to mature, and you can see that there is a lot of complexity in how this individual, how the entire organism changes. And we do these little plots not because they are beautiful, but because we want to know exactly why a cell decides to take one path or the other. And this is something that has been one of the most important development in the last couple of years, which is this organismal development, the ability to use super powerful computational programs to understand how from a stem cell we can actually derive so many different cell types and actually create an entire organism. So obviously, none of this would have been possible if we didn't have an explosion in the cell and the technology, and also in the throughput that we can get from this technology. So we started in mid 2009 when just only the ability to do a one single cell RNA-Seq to now with the being able to do millions of cells at once. And, obviously this has been an evolution of all these technologies. Every single technology has contributed to the evolution of another technology. This is when the technology brings more technology. And then we have all seen an explosion in the amount of cells that we can actually profile at a given time when we observe, when the microfluidic devices come up with something like Drop-seq, InDrop, or 10x Genomics that were able to sort of bring the number of cells that we can profile at once to in the number of thousands of tens of thousands. And in the last couple of years, what I've been, for me is, I'm a person that loves technology, and I like to see all these new papers and new technologies that comes around. You can see that there's been a tremendous amount of new technologies. There is every single something seq, and you can see that all of those some of them are actually very small changes from previous technologies, but some of them are extremely cool. One of the, you have cases like have changed the way that we actually do single cell profiling, like for example, site-seq, okay? And then you have all the situations in which you are able to perturb, to do CRISPR screens, you just modify some different changes, and that have allowed us to see the system or any profile in a different way. So, for example, now we are generating thousands of hundreds of atlases from different tissues, from different cell types, from different type of disease. So now we're moving now we need to perturb those systems. So now we are going to enter in another stage in which actually have atlases of perturbed systems. So, and all of these techniques here, they have contributed to that. They are contributing to that to be something real, okay? And I just want to highlight that this is just a few of the tech that has been coming up in the last year or two, but the list continues, and it's amazing to see, and it's very exciting to live in this time in which you see all of these things coming up. I would love to go over all of these, but unfortunately we don't have time. But this is an amazing, incredible amount of tech, and if you have time to have a look at those, I will strongly recommend. So now, in terms of application for single-cell RNA-seq, okay, so there is an obvious an underlying reason why we all do this is because we want to, exploit the ability to examine any transcriptional variation that may exist between individual cells. So it doesn't matter whether you are trying to identify or characterize a cell population, or you wanted to determine how heterogeneous that populations are, or to uncover new cell types and get some evidence of potential rare cell types, or whether you want to understand how the population changes over time, whether you want to see whether there is a sort of you want to understand the lineage and differentiation programs of a particular organism, or you want to see whether there is what's the nuances of all these interactions within the cellular pathways, understand gene regulation, something that we understand gene regulation in the past. We were thinking, okay, well, genes express and turn on and off. But then we've seen in the last few years, there'll be a few papers in that stochastic activation of genes, or seeing that not every cell actually expresses genes at the same time, which is, this is a fantastic discovery, and at least to me, it showed how little we knew before. And, you may want to characterize the immune profile, or you want to know how the tumors respond to therapy. It doesn't matter what you do. This is all of this. Some of your project will actually fall within this sort of basic or sort of high-level reasons why you may want to do single cell. Of course, you have the atlasing, which has been tremendous, have had tremendous success. But as mentioned before, this is something that now, once we got to that point, what we need to do is to start perturbing those atlases and see what new things come up. So now the point of this talk is not just to talk about all the other things that we already know. We already know how complex the system is. That's what you do in single cell. But I just wanted to highlight that the system can be more complex than we actually know or we think it is. So now when we do single cell RNA-Seq, there is always a challenge. And the challenge is that even though this is a technique that I've been, or a collection of techniques that have been around for 11, 13 years, it's still a new method. All right. So, and because it's new, it suffers from all the things that are new, and also it's changing over time. So something that you thought, oh, this is an amazing technique now, in a month or two, it may not be as good. Okay. So you will see that there was a lot of papers around with super cool techniques that will never be used. Okay. And so, and that's it because some of them are extremely challenging to implement. So some of the things that are very challenging is, they have to do with two parts. One is the wet lab, and the other is the dry lab. The dry lab will be all the informatics that are, just that allow us, the wet lab people like myself, to actually look at an issue from a different perspective. It's like, imagine you have a tissue that is like a Lego, and then you know that you have the Lego, you have different pieces, but if you can tease them apart, but these dry lab people, these bioinformaticians, as I like to call them, they allow us to see that every single piece can actually be observed in different way, and we can connect all these pieces in different way, and it's amazing. Okay, but there are challenges there, and there is a beautiful paper that is called the 11 grand challenges of single cell data science, which is not only taking into account current problems, but it takes into account also problems that may come, and I strongly recommend you to have a look at those, and again, this is just the only 11, but I'm sure there will be more coming in the future when we collect more and more data. Now, what we are going to talk today is essentially all the issues and potential challenges that you are going to be facing when you want to do single cell RNA-Seq and single nuclear RNA-Seq, or even if you do single cell attack-Seq or any multimodal analysis in the lab, in the wet lab, and the things that you need to consider from sample prep to sequencing. Okay. So it doesn't matter what type of experiment you're going to do, either if it's single cell RNA-Seq or single nuclear attack RNA-Seq or single nuclear attack-Seq or a combination of both. There are four major steps that you're going to be you're going to have to go through. So the first one is the sample processing and cell isolation. You're going to see them only in a little bit of detail. The second one is the cell barcoding and sequencing, which is essentially separation of the cell, individual cells. Then you are going to sequence that, and then you have to process. Okay, so you need to do all the mapping, the generating the count matrices and things like that. But then after that, you need to do the data analysis, which is mostly the interpretation. Okay. And we're not going to focus in the 3 and 4, but we're going to pay a lot of attention in the first 2. Okay. Now, the goal of the single cell RNA-Seq experiment, or the goal when you try to process your sample, is one, which is to prepare a live single cell suspension. And some people, the reason is, again, this is just my opinion. This by no means is a ground truth, but this is something that I've seen for a while. People prepare the samples, and sometimes they forget very basic stuff. Okay, like for example, and these are the high level considerations that I want you to bring to your attention, and things that you need to consider before you actually go into the lab. okay, and start processing these tissues. So one is that you need to consider the species that you are working with. It's not the same if you work with mouse or if you work with humans, if you work with zebrafish, because the way that you would process these tissues is different. And the way to preserve the viability, which is one of the main important part of this sample processing, is different. We have cases in which we had a fish, for example, that was from high salt content waters, and the cells will not survive the dissociation, okay? The cells will die. And they will die until we figured out why. We had many failed attempts, and one thing that we were not considering is the species. This species is a species that lives in high water content, so it has salt content water. So what we needed to do is just to maintain the high salt content until the very last moment, which was the encapsulation of that. So in this particular case, we even have to modify the chip to be able to work with this. But this is something that is important to consider, because if you forget about that, you may have a lot of failed experiments, okay? Now, the other thing that you need to consider is if it is a solid tissue, what organ? Are you working with brain, with breast, with liver, with lung? All of these tissues are different. They are different in terms of the extracellular matrix mainly, and they're also different in the way that you will target them. Some of them are more sensitive, some cells, some tissues obviously have different composition, and you need to consider that. And there is plenty of protocols available for different organs, and it's important that you don't just go blindly and try to dissociate the tissue with collagenase. That works, okay? So because it's exactly, this is a situation. One of the things that we need to consider is what are the composition of that tissue in terms of extracellular matrix. I will mention that a little bit in the talk. Now, if you're working with a liquid biopsy, for example, blood, that's okay. You know, this is a different you don't have to dissociate the tissue, but there is a lot of things that you need to consider from when you process this material. So from the blood draw to the time that it gets on the lab. There was recently a few papers. There was one from the group of Holger Hein, and there was another one a couple of days ago, maybe a week ago, about how sensitive is a transcriptome to all this time that goes from the sample processing to the sample draw to the lab. So that is part of the sample process. This is not separated from the sample processing, okay? So we need to consider that when we actually preparing our workflow. Now, the biopsy size or volume, here it's important that you consider here the volume and the size. It has nothing to do with the actual size. It has to do with the cellularity of the tissue. You may have a large volume, but it may be fatty, it may be fibrous, and what you want to know is how many cells you have in there, okay? So in terms of biopsy size, I get a lot of questions. For example, what's the best size to do this? And it doesn't matter the size, really. It matters how many cells you have in that little piece, okay? So it's important that you know that of your tissue. One thing that you will see if you're working with pancreas tissue, sometimes you have a lot of fat, all right? If you work with breast, you have a lot of fat as well. So you can consider that the same as when you work with brain, which is pure cells, right? Now, the cell composition is also another factor to take into account. And that is something that I keep insisting for people to know what the cell composition of the tissue is. I understand that we're doing this for atlasine, but there is a lot of work already on this. And it's sometimes what you can do is a pilot experiments in which you are determining the cell population using an orthogonal method. You can use FACS, you can do CyTOF, you can do anything you want to identify what's the cell composition. The reason why you do that is because you may be biased in your sample processing, and you're losing some of your cells. And if you don't do that, what you are doing is just biasing your single-cell RNA-Seq experiment. So it's important that you know what the composition of your tissues is, if you can, all right, as much as you can. Now, it's also related with the cellular heterogeneity. Not all the cells in the tissue are going to have or are going to be in the same transcriptional state, all right, or are going to have the same amount of transcripts or RNA in the cells. So some of them will have less RNA, some of these will have more RNA. And when you do the analysis, if you're only focusing those cells that with high level of RNA expressions, you may lose that. When we first started doing single nuclear RNA-Seq, one of the things that we noticed is that we were losing a lot of the lymphocytes. And of course, single nuclear RNA-Seq is a particular type of experiment in which you normally have less RNA. I will show you in a few slides later. But because we not consider the lymphocyte would actually have less RNA, we would actually filter them out. So this is something that you don't sort of take that into account during the processing, but it's important that you know what the composition is. That's what the composition is important here. Now, extracellular matrix composition is of absolute importance because it's what is going to determine what type of digestions you're going to use, okay? The extracellular matrix of a tumor may not be the same from a normal tissue, even from the same organ, okay? So, and the extracellular matrix from the brain is not the same from the lung. So you may use the most common enzymes, collagen enzyme, hyaluronidase, to do these associations, but it's important that you know that sometimes if you do that, you may be biasing the dissociation, the composition of your single cell suspension, because you may not be dissociating the cells of a particular group because the extracellular matrix was different there or require a little bit longer incubation time. So what you're doing is essentially removing the cells, some of your cells, as a debris because they don't dissociate well. So it's important that you check that, and because it can actually change the way that you do the dissociation. Again, there is a lot of protocols around there in which you actually can have a look and check. Now, if you are working with a post-mortem tissue, obviously you need to consider that because this post-mortem tissue usually are in the process of decay, so the cells are dying, and you're going to have a lot of cell death. So the way that you will process that sample is not going to be the same if it's a freshly live. You probably have to go through some sort of cell death removal. I'm going to discuss that a little bit as well because I'm not really keen on the cell removal, and I will explain the reasons why later. But you may decide what type of process you're going to do. Maybe you want to avoid further death, and you may want to go to single nuclear RNA-Seq. Okay, we're going to discuss that again a little bit later. Now, if you're working with a naive or chemotherapy-treated tumor, that it's, for example, is another situation in which you may decide to process in one way or another because, a chemotherapy-treated tumor, if the therapy is a cytotoxic, you may have a lot of cell death, and that's going to completely ruin your experiment. Because what we really wanted to know, remember, is to have a cell that is, cell suspension is highly viable, okay? And also, you may consider the way that you process your tissue had to do with how you preserve the tissue. Okay, it's not the same if you preserve the tissue using if you use cryopreservation or if you snap froze the tissue. So if you use cryopreservation, you can dissociate the tissues in the normal way. You can just do a few washes and then proceed through the dissociation after you use the right enzymes and procedure. But if you do snap freezing, you're not going to snap freezing as in flash freezing, just put the tissue in liquid nitrogen. That is not going to, you're not going to have any live cells there. You're probably going to have chunk. So, and that is going to change the way that you process the tissue, and it's likely you're going to have to go to single nuclear RNA-Seq. So now, in terms of dissociation and the way that you will actually decide how to proceed, there are these three beautiful papers that I absolutely recommend to go and have a look. These are probably my favorite in this sort of area. This from the group of Samuel Aparicio and Saurabh Shah. So this is from Kiara O'Flanagan. See, this is a very nice paper. We are going to discuss a few of these results here, in which they sort of introduce the cold active protease to do dissociation instead of collagenase digestion, 37 degrees. And there's also used here in the Elena Dizenko, and this is a colleague of mine in Australia, Alistair Forrest. So they had the RNA, again, a beautiful paper. These two are complement to each other. They look at the problem in different angles. Some things are very similar. They found very similar results, but they both conclude the same situation. Cold-active protease is a very good alternative to, actually much better alternative than collagenase digestion. And then we have this, the one that I mentioned before from Ramon Barroso. So this is from the group of Hauger-Hein. This actually, they take into account the processing of blood, and how you need to all the things that you need to consider from the moment that you take the sample draw to the moment that you process, and how the transcriptome and the epigenome may be changed. So if you have time, and if you don't have time, just make the time, have a look at this, because these are absolutely amazing papers to have in your mind. Now, and the other thing that also going to change the way that you process the tissue is whether you want to work with all the cells in the tissue, or you have just only a group of cells that you are interested. You may only be interested in CD45 positive cells, or maybe you just only want B cells or T cells or tumor cells, or depending on the ploidy of the cell. So in this case, you have to introduce some sort of FACS sorting, and also that changes the way that you process the tissue. Okay, now usually the way that you process the tissue mechanically is the same, because you need to do single cells for FACS sorting, but there is another step that you introduce, and that is another step in which you may introduce some stress. Okay, we will discuss later. Now one of the things is that I continue sort of insisting here is that there are protocols available for dissociation from pretty much every part of the body. And I would recommend to, before you just going blind and just use the some X lab did, you do your pilot, you take that protocol, protocols.io, it has plenty of protocols available there, and just get in touch with the researchers, talk to them. Don't be afraid of talking to these people, because they will actually give you the tips. Some things cannot be written in papers, okay? And we all know that papers will only have the things that are successful, but there are a lot of things that are not successful. So discuss with them, just get the time in this Zoom era, so make the time to talk to them and tell you to give you all these little nitty-gritty tips that are going to be so important for the next stages of your single-cell RNA-Seq, which is generating live single-cell suspension. Now, of course, the methodology and the technology that you're going to use, whether using droplet or plate, may have an impact in the way that you will choose to process your samples, and that it's also very important that you take it into account. Now, when you make the decision, again, this is not technically related to sample processing, but it's something that I want to put out there up front. When you make the decision of what type of technology you're going to use, make sure that you do an informed decision. Just don't use the one that is in fashion at the moment, because it's great, and Lab A actually had a lot of success. There are very beautiful benchmarking studies, again by the group of Hauger-Hein and the Levin Lab from here at the Harvard and the Broad Institute, in which they have spent thousands of dollars doing comparisons across different platforms to give you, not just to publish Nature papers, but it's just to give you an idea of what is, they are making it easy for you. Just, these are the techniques, these are the ways that you need to be looking. And if you want gene expression, if you want, clusterability, mappability, what is that you're actually focusing here? Just make sure that you take these papers, you read them, and decide what technology is more suitable to you, okay? I understand there are some technologies that are more common and famous and more easy to use, which we also discussed, but it's important that you do an informed decision before you actually choose the experiment, because it actually can save you a lot of money, okay, and headaches. And finally, if you want to do joint profiling, for instance, if you want to do, single cell attack-seq, or single nuclear attack-seq and single nuclear RNA-seq, you together, or simultaneous, if you like, you cannot prepare the nuclei in the same way, okay? You cannot prepare the cells in the same way. There will be some things that are shared, but then you need to separate. For example, when you do single nuclear attack-seq, you need to permeabilize the nuclei. So if you permeabilize the nuclei, you're going to lose RNA, okay? Some people have shown that in recent papers that they don't lose a lot of RNA, but there is a lot of loss of RNA. I have seen it, at least, as I say, this is my experience. We've seen loss of RNA when we permeabilize. And then you end up having, you already have a little RNA in the nucleus. So if you keep permeabilizing, you keep leaking a lot of RNA, you get a lot of background, especially if you're working on a droplet-based approach. So that would, again, whether you're doing a joint profiling or no, it will actually change the way that you prepare your single cell suspension. Now, the second part, and just the second part of the process, which is the cell barcoding and sequencing. So here again, the same situation. The first thing that happens is when you actually capture the cells and you kind of individualize the cell. And this is an important part because once the cells is individualized, that happened in the previous case, there is something that it's very common in all these applications. You need to essentially individualize the transcript, and you need to make sure that when you barcode, you label that barcode, we know informatically that comes from cell 1 or cell 2 or cell 3. Okay, so in all of these systems, the cell barcoding happens pretty much in the same way. There are slightly differences, but the first thing that happens is a cell lysis. So you lyse the cell and the RNA gets released. Whether that happens in a droplet or it happens in a well. Then all of them use an oligo-DT. Okay, and that oligo-DT sometimes, in the case of, for example, of 10X, is in a dissolvable bead. In the case of Drop-seq, is on a solid bead. And in some others also it's a solid bead. And in case of in-drop, for example, the release, in the case of Drop-seq, there is no release of the oligo. In the case of 10X, there is release of the oligo. And in case of in-drop, there is release of the oligo. But the release of the oligo is via UV cleavage of the oligo. And then the oligo gets released and sort of capture the messenger RNA through the poly-A. Again, that can happen in droplet or in well. And after that, you get that first strand synthesis. That's a famous reverse transcription step, right? Now, in some cases, you get the reverse transcription in the droplet or in the well, like in the case of 10X. In some cases, you actually do the reverse transcription in mass. So, for example, in case of Drop-seq, it's only for a tiny little period of time in which the RNA is in the droplet, and the beads are in the droplet. So you capture the RNA with the poly-T, and then you just break the droplet, and the non-transcribed RNA, it just pull all together, and then it's transcribed in mass. Okay, just subtle differences, which may or may not have a high impact in the quality of the data. This is not what we're discussing here, but different ways to do sort of the same thing. Then you have in some instruments, some technologies, you have what is known as a template switching. I will discuss that in a little bit, what it is, but it's essentially, it's a way of creating a docking site for a primer for cDNA amplification. In other approaches, like in-drop, you don't have a template switching, okay? So what you do in the case of in-drop is something different for cDNA amplification. And in the case of Quad-seq, for example, you get an extension of a poly-A in which sort of like changes the way that you do the amplification. So you create the docking site using a TDT, not a poly-A, a poly-T, and then that is what is used as a docking site for a primer for cDNA amplification. Now then, You may or may not have a second strand synthesis. That depends on the method. In the case of 10X, for example, you don't have a second strand synthesis per se. You go straight into the amplification, but in other technologies you do, and then you do a cDNA amplification. In the case of 10X, you have a PCR amplification that we already know. In the case of, probably if you're not very familiar with, the case of InDrop, the standard InDrop, we actually currently here at the lab, we have modified the InDrop to actually do what is called switch drop, which essentially it's basically the same approach that is used 10X, sort of smart-seq approach, but it's not the standard InDrop. But in the standard InDrop, what you do have is, once you have the cDNA as a hybrid, what we do is a second strand. We do an in vitro transcription, and then from that in vitro transcription, that's why we amplify the RNA, and after that we do reverse transcription again. It's a little bit cumbersome, but actually you can generate single cell data of high quality as well. And after that, that's how you do the amplification. And from that, you have to fragment the material. In the case of 10X, you can use enzymatic fermentation. Some others use mechanical fermentation. In the case of InDrop, because you are doing an in vitro transcription and then you do a reverse transcription, the fermentation happens it's not mechanical, it's not enzymatic. It's happened because you're using random priming that it binds on different pieces and different places of the cDNA. And after that, you make the most important part for the next stage, which is the library preparation. Now, the library preparation can be a full length, okay, entire transcript, or you can be 3' or 5' bias. And the reason why you choose one or the other depends on what you want to do. Okay, sometimes you want to just only detect things that happen in the 3' or just the 5', and you want to count just molecules, just determine the number of molecules that is between this cell type and the other cell type, and between this transcript and the other transcript. But you don't really you don't care about potential rearrangement that may occur in the transcript. For example, fusion genes or splice isoforms. So you just don't worry about those. Okay, and in some other situations you do need, and that's why you need to do full length. Again, not all of them allow you to do full length. For example, 10X you can do full length, even though you have a full length cDNA after the cDNA amplification. You don't do that. For that, you can do something like smart-seq if you want to do full length. But obviously, a library that is a full-length library is going to cost you more in the sequencing, but it has huge amount of advantages as well. And after that, you go in the sequencing step, which is a very important part of everything. Now, before I'm going to briefly mention what are the different technology options that are available, and then I will stop this part of the talk here, and then I'm going to resume in about an hour, so we can have a little bit of discussion here if you have any questions. I cover a lot of parts here, so in the second part I will cover a little bit of these technology options. But just as a small teaser, there are several technology options that are available for doing single cell. One of them is the one that you can do in wells. Usually they are assisted by either fax, or it can also be assisted by a liquid handler. Okay, the most common is assisted by fax, and then you have cases like within this system you have Cell-seq, Mar-seq, Scrub-seq, Quad-seq, but and then you have Smart-seq. I'm going to mention a little bit more Smart-seq because it's the one that has been sort of like the grandfather of all these other approaches, and also because it allows you to do something that not all the other techniques allow you to do. Then you have something like in nano wells, like in the case of Seq well, and you have micro wells in the case of BD Rhapsody, and then you have the most commonly used at the moment are the things that are done in droplets, in which we have chromium, obviously, and we have in-drop, and we have drop-seq. So in the next second part of the talk, I will just briefly describe a little bit of this with a little bit more details, comparing these technologies, and also getting to some more juicy parts related to also how to keep the preservation of your tissue and the dissociation of your tissue, and how to improve the preparation, and some things related to experimental design. So if you have any questions, this is a good time to ask, and then we can resume in about an hour.
Thank you, Luciano, for that outstanding presentation. We will now move into the live Q&A portion of the presentation. As a reminder, please submit your questions in the Q&A box. So our first question is, where do you see single cell and single nuclei RNA-Seq developing in the future?
Oh, wow. Okay. Look, I will discuss a little bit briefly the differences between these two, and I think that these are, they are not mutually exclusive. What we do have is situations in which we may have the samples that are more amenable for a particular type of sequence of process than others. And I think one of the things that I like about single nuclear RNA-Seq in particular is that it allows you to recover those samples that sometimes you cannot process using single cell, especially those that are being frozen. But I will definitely discuss this in the second part of the talk.
Okay, thank you. Our next question, what's the best way to preserve cell viability?
There is not a single way to do that. I think essentially you need to know that your cells are, the moment that you dissociate them, they are not happy. Okay, so you need to keep them, or try to keep them at 4 degrees as much as you can. But again, you need to know what is the best media that you can preserve them. There is not a straight answer for everything. So I think that's depending on, it's very sample dependent, and there is no one better than yourselves to know what's the best condition for your sample. And that is something that you probably can gather from running some pilots on your samples. But there is not a recipe. I know people want recipes. This is the best way to do it, but in reality, this is not what happens.
Okay, thank you, Luciano. Our next question is, why is cell viability so important?
Well, the cell viability is important because when the cells are dying, okay? So imagine you have a cell suspension. When the cells are dying, a few things happen. So the first thing that the cells is going to do is just to lose this impermeability of the plasma membrane. And as that happens, you start losing RNA out of the cells. And when you lose RNA out of the cell, that RNA, if it is captured in a droplet, for example, so that RNA is going to go into the reverse transcription mix. Okay, and when that is captured in the droplet, you are introducing background to the actual analysis. Okay, now the other thing that happens is that because the RNA comes out, you get and mitochondria cannot go out because this is a massive structure compared to the holes that may be created on the membrane. What you end up having is a lot of mitochondrial rates, and that is actually pasting your data. So it can be useful for some approaches, but usually this is not what you want. Okay. Another thing that you will observe is that other RNAs that are not able to leave the cells are these long non-coding RNAs that are located in the nucleus. They're usually trapped in there. So what you end up having is a lot of mitochondrial rates, very low RNA or feature counts, and a lot of low UMI counts. And when you do have, that's what is diagnosed as a bad quality cells, and those cells are going to be removed. So you end up having wasting a lot of data sequencing in cells that eventually, or what we call barcodes, that eventually are going to be removed because they are of bad quality. And of course, when you have that particular background, that is a badge effect as well on itself. Because then if you want to introduce that, if you want to compare those cells with another, this sample with another sample that has been processed properly, that background is part of your, your analysis, and that can actually also introduce. You will have to correct for that because that is introducing variability across the two data sets. Aside of the fact that they are already run differently. So all of that contributes to potentially making your data analysis more difficult, and your bioinformatician is not going to be happy with that.
Okay, thank you. Got another question. When would you use FACS or MACS?
I will discuss that in the second part of the talk. This is, this is an important aspect, but I will just go through the slides in the second part. So if you guys stayed, I will definitely describe when is better and when is not. Okay.
And replicates, what's your opinion?
Again, this is something that comes in the second part of the talk.
Okay. One last question here. What is the most important factor for successful single-cell sequencing outcomes?
Well, there are not really unique single-cell unique aspect. I think probably cell viability is the thing that I would try to preserve in every single time and try not to stress the tissue much. But again, there is not a particular thing. That's why this talk has so many different parts, because every single part actually contributes to a high quality data sets.
Well, thank you so much, Luciano. We're out of time for today, but don't worry, audience, any questions that are submitted and not answered today by our speaker will be addressed via email. Thank you again, Luciano, and thank you to the audience for those outstanding questions. This presentation will be available for on-demand viewing for 12 months. Please remember to share it with colleagues who might be interested in today's topic. And don't miss out on the other presentations on our agenda. Visit the Agenda tab in the auditorium for a full listing. Thank you again for your participation. Until next time, goodbye.